So at the end of last class, we kind of got into the details of phototransduction, and we saw that it's not straightforward. So, how well, we should say, what is the cell that actually does the phototransduction? What cell is actually responsible for converting light into an electrical signal?
A rod or a cone.
Yeah, exactly. Rods and cones. So then the question is, how does a rod or a cone convert light, which is, again, it's just electromagnetic radiation. It's just this weird energy, into some electrical signal. So hopefully you guys have spent some time also over the weekend going back over that and making it make sense. So, like, what are the steps? What happens here? So if we have, let's say we have light come in, I'll make it light yellow. So light's coming in. What is light interacting with in the rods and cones that's gonna help start this whole process?
There's the opsin that has the retinol on it that's made from vitamin A.
Exactly. So let's say this is our rod here. And in the membrane of this, what we call the outer segment, there's the opsin, which is a membrane protein, plus retinol, which is based on vitamin A. And retinol has different names. Sometimes you can be called retinine. And in rods, sometimes you call this rhodopsin. Again, in the old days, they would even call it visual purple, because before they had any idea what it was, they knew that it looked purple and then bleached out in light. But again, when that light hits, it gets absorbed by this opsin and retinol, and then what happens?
The retinol changes its shape.
Yeah, exactly. So the retinol, which is normally in this kind of cis-retinol, kind of bent two sides of the molecule the same direction, goes to the trans-retinol, where the two sides are bent opposite ways. And again, that's going to activate the opsin. This is kind of the equivalent of a receptor binding a ligand. Normally, like when we think of signaling in the cells with these membrane proteins, you've got some, there's some membrane protein that's a receptor with receptor, and there's some ligand, and there's like this lock and key, the binding. That's kind of what we've been thinking of. But instead, we've got this situation where the light transforming the retinol is going to activate the opsin, rather than having kind of a receptor getting activated by binding a ligand. But the next step is just going to be activating a G protein. And the G protein in this case has a special name. If you want to call it transducin, you can call it that, or you can just call it G protein. What happens? So remember, when we act in general, what happens when a G protein gets activated? G protein is the first step in initiating what?
Messenger pathways.
Exactly. So this is going to activate a second messenger pathway. You definitely want to remember second messenger pathways and all that till the very end. I mean, the last week of this class, we're going to be doing like the endocrine system, and that's all about hormones are just ligands, attaching to receptors and initiating second messenger pathways. This at the core of so much of how the body works and how cells sense the world around them, how they talk to each other, how they coordinate all their actions. So second messenger pathways, hopefully one of the things you walk away from this class with is having some basic sense of like, oh, I know what a second messenger pathway is. In this case, the second messenger pathway is going to start a whole chain of events that's going to ultimately turn on phosphodiesterase down in the cell. What does phosphodiesterase do? PDE. We met that like again the first week of class.
It breaks down the cGMP that holds the sodium channels open.
So that's it. So this is going to break down the cyclic GMP. And that cyclic GMP was part of the pathway that was actually holding the sodium channels open down here. So we're going to close the sodium channels. And what is going to happen to the membrane potential when we close sodium channels?
It goes down.
Exactly. VM is going to go down. So that is signal transduction, right? The whole point of what we're talking about here, how does a rod or cone convert light into an electrical signal? If just saying, like on an exam question, there is some question about like how does your body accomplish phototransduction? How do we convert light into an electrical signal? That is the real meat and potatoes of all the other stuff that we talked about in the eye with the lens and changing the curvature and focusing. That's so we actually don't have a blurry image. But that's not actually about the sensory transduction. The actual converting the stimulus into an electrical signal is all the stuff that we've just been showing that's happening in the specific rods and cones. So this is phototransduction or sensory transduction in the eye to convert light into electrical signals. And then we talked about how the next steps, if you wanted to get into more detail, rods and cones are kind of in your retina. Cones. And then if their voltage is going up, then it's going to open calcium channels, which means that they're going to be releasing glutamate. Right, so more or less glutamate is going to be depending on more or less light. And then these are going to talk to the next level of cells, which we call the bipolar cells. These are going to talk to the next layer of cells, which we call the ganglion cells, that actually have axons that make the optic nerve. Again, everything we're talking about here is the retina. There's other cells in the retina. There are what are called horizontal cells that are going in here. There are amacrine cells that are in here. Like I mentioned before, the retina actually originates from the diencephalon. So it's like a little outpocketing of your brain. So there's a lot of kind of interesting, complicated processing going on that we are not going to cover. I can say I took an entire semester just looking at the retina. There's a lot of interesting pre-processing going on, kind of sharpening images and boundaries and this and that. But that's for your, for further, if you go deeper into vision, you'll learn about that. The last thing I want to talk about before we leave the eye is kind of how your eyeballs, or how this optic nerve kind of heads into the brain. And vision is a little different from the other senses. We've been talking about, in general, there's this lateralization. Like if you think about, say this is your brain. Let's let this is you with your ears and your eyes. This is obviously kind of looking at you from the top. Normally, let me try to figure this. Which side of the body is controlled by which side of the brain in general?
The opposite controls the opposite side.
Exactly. Which again is really important, right? If you have a stroke that damages your left primary motor cortex, it's your right side of your body that is going to end up getting paralyzed. So in general, there is this connection of the one side of the body to the other side of the brain. With your eyeballs, it's a little different. The way your brain and your visual cortex is connected to your eyes is not about side of the body. It's about side of your visual field. So, for instance, things that are on this side of the body are going to be kind of going to this side of the retina. Things that are on the other side of your body are going to be going to the other side of your retina. And what happens is each eye sends information about the side of your visual field to the opposite side of the brain. So like this is going to go here, is going to go up into its opposite side. It's still opposite side, but it's opposite side of your visual field. So both eyes send information about that side of the visual field to this visual cortex. Same thing with the opposite visual field. Both eyes are going to send information to the brain about that side of the visual field. So when you actually, in anatomy, I'm sure you remember, if you actually look at where the optic nerves enter the brain, they create what's called the optic chiasma. Let me slow down here. if you look at like the optic nerves coming in from each eye, they kind of just create this kind of X and continue on into the brain. But in reality, what's happening is, again, you've got each eyeball is sending stuff to both sides of the brain, depending, to give the brain information about the specific side of your visual field. And we saw in lab, why does it make sense to actually get information from two different eyes about the same part of the world? Right? This visual cortex here is getting information about basically the same part of the world from two different eyes. Why is that useful? Why not just have one eye? Or why not? Remember, we talked about if you were more of a prey animal, if you were like a mouse. You ever look at a mouse? Their eyes are kind of on the side of their head here, and the visual field for a mouse is basically 360. Like, you can't freaking sneak up behind this mouse because it can see behind itself, basically. I mean, more than that, mice actually have pretty horrible vision. They rely on other senses as well. They have really good hearing. They hear frequencies way above what we would hear. Whiskers are actually amazing in terms of giving them a whole other, what we call umwelt, kind of way of knowing the world, that's very three-dimensional, the way. So they use their whiskers as a whole other sense that we can't really quite imagine what it feels like. But in terms of pure vision, they have two eyes, but they use them to overlap. I mean, not to overlap, to actually give them 360 vision. What is the advantage of having two eyes that are kind of looking at the same part of space?
Depth perception.
Yeah, this is gonna, your brain basically does some calculation that's going to give you depth perception. We call it like stereoscopic vision. There's, again, and we saw in lab, there are other things that give you a sense of depth, whether or not some, perspective, things get smaller as they get farther away. That gives you an idea of the distance. Things block each other, right? Just because someone only has one eye doesn't mean that they're not allowed to ever drive even, but they got to be kind of more careful because you don't have this real sense of depth perception that you do with two eyes. So any questions or anything about the eye before we move on?
The same signal transduction process happens in both rods and cones, yeah?
So it is the same process. The difference is the opsin. Depending on which opsin it is, you can tune it to absorb light at different frequencies. So with the rods, they absorb light kind of optimally around in this green zone, kind of spread out. For the cones, it's more kind of tightly tuned and specifically just around red for the red cones, just around blue for the blue cones, just around green for the green cones. But the basic process is the same. The other difference is, I need to erase some of this stuff here, clear all the drawings. if you remember, if you're looking at like the eye here and the lens and the retina, here's light coming in. The cones, which tend to be in that fovea centralis, tend to be connected to the ganglion cells. So each cone gets its own axon. It's not coming out of the cone, but if you look at the wiring from the cone to the bipolar to the axon, each axon from the optic nerve is basically responding to input from a single cone. So that gives you this real nice spatial resolution, but it means you got to really put a lot more input into this cone to get a big enough signal to actually trigger the process, which is why the cones need brighter lights to have a response. When you're looking at rods, which are more on the periphery, you tend to have a lot of convergence, lots of rods coming together to become one signal going out, which is why you don't have spatial resolution. You can't really tell where exactly where the light is falling, but because it's integrating across many rods, you can have a much dimmer light and still ultimately integrate, have a large enough signal to say, oh, there's at least something there. So even though they're both using the opsin and the retinal, the rods, because of the way they're kind of wired up, end up being much more sensitive to dim light, where the cones need brighter light. But it's not about the core process that's happening. It's about kind of how you use those photoreceptors and how you wire them up and all of that. So cool. All right, so we are now going to move on to the ear. What? Oh, I want to clear all drawings. Wait. All right, how do I get rid of all these? Oh, no, all right. So I've never got to this place before. All right, so the ear. So first thing to talk about with the ear is the ear is doing lots of things. Again, we think about it doing hearing. That is just one thing it does. This is going to be the cochlea. Cochlea, which actually means, comes from Latin for snail. It actually kind of looks like that. So hearing, we're going to spend a lot of time talking about hearing, but that's just one part, the cochlea. There's also a lot of equilibrium senses. Equilibrium and balance. We're going to talk about the saccule and the utricle. So like 2 and 3, this is going to be about which way is gravity going. We're going to talk about the semicircular canals. There's 3 of them. Let's say 4, 5, 6. So there's like 6 different parts of the ear that we're going to talk about. 5 of which actually have nothing to do with hearing, are actually about equilibrium, about which way is gravity going, which way is your head spinning. So hearing, equilibrium, and balance. We're going to look at all of these. And to introduce this, all of them, the thing they all have in common is they all ultimately rely on mechanotransduction, kind of having some physical movement that then creates an electrical signal. If we're talking about hearing, the mechanical movement is ultimately the vibration of your eardrum, which is going to get converted into electrical signals that go to your brain and you perceive as sound. If we're talking about like, your dynamic equilibrium, it's the moving of your head is going to cause currents in this fluid that bend this little membrane that is going to ultimately be picked up as or turned into an electrical signal that is then going to go into your brain and be perceived as I'm spinning. So let us introduce the hair cell. So yeah, I wish I knew how to get rid of these whiteboards, but I don't. Okay. So the hair cell. Which I actually have, I can say I have kind of an affinity for this hair cell. This is what I actually spent like 5 years of my life studying in grad school, basically, was a lot of the details and dynamics of hair cells in the ear and how they interact with the membranes overlying them and how they work to both do the transduction as well as how they then help pull information from that movement, like the frequency, how fast is it going, or when did it start, when did it stop, and all that. I'll talk about that in more detail later. But ultimately, whether you're talking about hearing or whether you're talking about balance, it's ultimately going to rely on the hair cell. So let's introduce the hair cell. It's called the hair cell because it has these, remember cilia? So it's got this one true cilia, they call it the kinocilium. And then these other parts, they're actually not true cilia. They're actually what we call stereocilia, meaning they don't have the microtubule structures inside, but it doesn't matter. So this whole part up here is called the hair bundle. Right? It's not really hair. It's just little like extensions of cell membrane with cytoskeletal components holding them up. And then there are these things that are called tip links. There's these little tip links. And they're attached to little membrane proteins that are actually little channels and stuff in there. And then typically a hair cell then has a neuron that is connected to its bottom that it can use to send messages, to talk about what's happening. The basic idea, this is a mechanoreceptor cell. So mechanoreceptor meaning mechanoreceptor. Mechanoreceptor meaning it takes a physical movement and converts it into an electrical signal. In the terms of the hair cell, if you take this hair bundle and you bend it towards the kinocilium side, this will depolarize the cell. If you take the hair bundle and you bend it away from the kinocilium, this will hyperpolarize the cell. So VM goes down. If you bend it the other way, VM is going up. So this is mechanoreception, right? Oh my God, mechanorector. People should tell me if I am a total spaz up here, man. I'm like, because this is getting recorded, so then it's even more embarrassing for me that some random person's gonna say, this guy can't spell. Um, Mechanoreceptor. So it's a mechanoreceptor. You bend this hair bundle in one direction, the cell becomes more positive. You bend it in the other way, it becomes more negative. That's sensory transduction going on. As the membrane voltage goes up and down, it's basically going to change the rate of action potentials that are going to be sent down this neuron here. So I can tell you that it's very, like, again, when I used to stick electrodes in the auditory nerves of frogs, if you excite these things more, you can just hear more action potentials going down fast. And when there's not much of a stimulus, when these are not getting, you don't have the action potentials going down. So basically, as you bend this in one direction, voltage goes up, which is going to bring this thing closer to its threshold, which is going to make it more likely to be sending down action potentials to the next levels of processing. You bend it the other way, it is then becoming less excitable, less likely to fire off action potentials. So that is going to be the core. Whether we're talking about hearing or equilibrium and balance, the hair cell is going to be the kind of the rock star that is going to be ultimately giving us some electrical signal to send to the brain. Obviously, though, the stimulus is going to be different in the different types, whether we're talking about balance or hearing. So the other parts that go from stimulus ultimately to a hair cell bending are going to be important. So that's what we're going to look at right now. Before we do that even, we got to kind of look at the ear as a kind of bigger organ. So let me bring up a picture here.
So it has to do with the kinocilium bending?
Yeah, it has to do with the whole hair bundle bending. These little tip links, as you kind of stress it in one direction versus relax it in the other direction, actually is responsible for opening and closing the little channels that are creating these changes in membrane voltage. So, all right, so now I'm going into my physiology lecture images, which I just created. So I'm going to go share screen. All right, you should all see, this is just something I found on the internet. It's basically a picture of a hair cell. And this is kind of just showing that when you bend it in one direction, it will depolarize. And as it depolarizes, it will then kind of excite the next neuron and send messages. When you bend it the other way, the hyperpolarization, the voltage goes down and it stops sending the message down that axon that's connected to it. So, again, beyond that, don't worry about the details. The actual voltages in the ear are actually fascinating, but beyond what we have time to look at. Normally, we think about sodium coming in to make things positive, but the ion concentrations in the fluids in the ear, the endolymph and perilymph, are really different from what we normally think of as intra and extracellular ion concentrations. So if you start reading more detail, you'll realize, oh, it's kind of weird. Normally, if sodium rushes in, that's actually going to make something more negative. But in this case, it's actually doing the opposite because the ion concentrations are really different in the fluids inside the inner ear. So don't, yeah, so for the purposes of our class, don't worry about the details of the voltages. Just know that when you bend it towards the kinocilium, it becomes more positive and we get more signals going down the neuron. When you bend it away, the voltage becomes more negative and you have less signals. All right. Now, back. Mouse, back to here. Ah, clear. All right, so anatomy of the ear. So we saw this briefly when we had our lab. We talked about how there's this outer ear. Let me use a color that's not the same color as everything else here. There's the outer ear, which is basically where the sound's coming in. You actually do, we care about it a little. For our class, we're not going to care about it much. Other non-human animals, again, if you guys have cats and dogs, you've probably all seen that, like, their ears are actually, I guess here, I'm going to turn off I think in order to actually show this, I'm going to, you'll get to see my messy office here, but I need to be able to kind of show you. So when they have their ears, if you look at a cat and you like, the ears totally go towards where you're doing, and they use their ears for their emotions, like nonverbal communication. It's like they're pissed at you. They got the airplane ears, so it's like for a lot of non-human animals, the external ear is actually kind of useful for lots of things and are more interesting than ours. For us, we'll just think of it kind of like a little funnel to bring the sound in. So, and we're not going to look at the external ear beyond that. Then the ear canal is heading on, and everything else now that we're going to look at is all inside the temporal bone of your skull. So all this stuff here, this is in the temporal bone. In the temporal bone. So this continues to all be bone. So there are two important little caves you need to be paying attention to. One is the little cave that we call the middle ear. So the middle ear is right here. And then there's the inner ear. Which is all this stuff over here. The inner ear, this is going to be where you actually have sensory transduction going on. This is where the hair cells are. So this is but this is all fluid filled. I wonder if I can, text, holy crap, I can use a little text. Let me see how this works. This is fluid filled, woo! So inner ear is fluid filled. That's where the hair cells are. Obviously the sound that's coming in is air vibrations. Sound. So air vibrations And yet we want to bend these hair cells that are sitting in this fluid-filled place. In engineering, we talk about impedance matching. If you have two kind of different systems and you want to efficiently transfer the energy from one into another, you actually have to be kind of intentional about it. If you just took kind of the air vibrations of sound waves and kind of pushed them up against the little membrane on the inner ear, most of the energy would just bounce back. You wouldn't efficiently transfer the air vibration into vibrations in this fluid-filled inner ear. So that's kind of what's going on with the middle ear here. The middle ear is going to be a way to efficiently convert air vibrations and movements into putting vibrations into the fluid of the inner ear. So let's start by looking at the middle ear here. The middle ear is going to have its entrance as the tympanic membrane, which is basically your eardrum. So as the sound is coming in, which is just vibrating back and forth, the eardrum vibrates back and forth. You know, it's just this little membrane. It's connective tissue on one side, epithelial on the other side here. And then there are these three little bones, malleus, the incus, and the stapes, also known as the hammer, the anvil, and the stirrup. Stirrup because it actually looks like a little stirrup. And then there's this little membrane right here that the stirrup grabs onto that is going to actually put the vibrations into the inner ear. It's going to be where you have the oval window. So actually, let me go and so here we had the outer ear going in. Here's the middle ear. And then this is the inner ear. Again, this is all in the temporal bone here. Here's the eardrum, or the tympanic membrane. And those little ossicles, again, like that hammer, the anvil, and the stirrup. They're itsy bitsy bones. They are the smallest little bones in your body. They actually have tiny little synovial joints. So as long as everything is working properly, they should all be able to move back and forth, kind of like a little lever system. And what they do is basically efficiently transfer the movement, the vibration of your eardrum, which is moving due to the sound. And they push and pull on this little thing here, again, which is the oval window. The oval window, which is the entrance into the inner ear. Again, the inner ear is fluid filled. And again, because fluid is not compressible. If you have a thing full of fluid and you try to squeeze it, it doesn't really squeeze. So if you want to be able to push and pull on this oval window, you actually need to have something to relieve. If you push in, you actually need something that actually pushes out a bit. So this little thing that allows you to push and pull on the oval window it's going to actually basically do the opposite. It's called the round window here. right? So if you push in on the oval window, the round window will pooch out a bit. If you're pulling back on the oval window, the round window will suck in a little bit. So as you push and pull on the oval window, you're going to be creating these waves in the fluid of the inner ear. We're going to come back to the inner ear in a few moments, but let's keep our attention in the middle ear just for a few moments. So again, the ossicles are all about being able to efficiently transfer the energy from the air vibration into a vibration in the fluid-filled world of the inner ear. Conduction deafness, which we talked about in lab a bunch, right? That is if this process is not working. It could be because the ossicles aren't moving. Like my mom's hearing was really bad because she had these bones fusing, so they didn't move. And so if these bones didn't move, it didn't matter how much the eardrum was moving. There was no single signal getting sent to the inner ear, which is where the hair cells are, where the signal transduction happens. Conduction deafness is if you burst your tympanic membrane. You have a burst eardrum. Obviously, if your eardrum's burst, the speaker cone's gone. That's going to not be very good in terms of transferring energy from the air vibration into the inner ear. You can have otitis media, like inflammation in here, all this fluid filled up in here where the things don't vibrate well. There's all sorts of things that can go wrong. Lots of earwax, right? You know, you don't have the vibration coming in because it's not even getting to the tympanic membrane. So middle ear, you actually have a couple of muscles. There's one that actually grabs onto the stapes, the stirrup bone. There's another one that grabs onto the hammer. It's called tensor tympani. So there's actually these two muscles in here. I'm going to like erase part of it. Here's the ear, but I just want to give myself more room to write. So there's, oh, I didn't turn on my little extra keyboard, which I'm doing now. There's tensor tympanum muscle, which is attaching to the hammer part. There's the stapedius muscle. Which is connecting to the stirrup. So these two little puppies can actually tense up if your body wants to reduce the amount of energy that's getting transmitted to the inner ear. So if there's really loud sounds, these muscles will tense up, and it's a way of protecting the little hair cells so you don't overstimulate them. So this is one of the ways your body has, like think about like if the lights are really bright, the pupil gets really small, the iris constricts, so you have less light coming in. If the sound is really loud, these little muscles are going to tense up, make sure these ossicles don't move as much, which means less of that energy sound energy is going on into the inner ear. The one other thing we need to talk about the middle ear before we leave it alone is, you know, it's closed off on this side, it's closed off on this side. You know, if there is whatever the air pressure is in the outside world here, maybe let's, yeah, there's some air pressure out here. You want to make sure, and that's pushing in on the eardrum, you want to make sure that the pressure on the other side of the eardrum is balanced, or else your eardrum's going to explode or implode. So we need to balance the air pressure on either side of the eardrum. If you don't yeah, then again, minimally the eardrum gets kind of stretched and is not going to work so well, but it gets more and more painful, and then it literally can burst. So we need some way to adjust the air pressure inside the middle ear to balance the air pressure on the outside of the eardrum. That is going to be using the Eustachian tube. So there is actually, I need to like kind of make this not as complete here. There's actually a little tunnel that comes up. This is my Eustachian tube here. Also called auditory tube. It also has other names like the pharyngo-tympanic tube. It basically goes to the back of the throat. Up into what we call the nasopharynx. In fact, I can go and share screen. Doo-be-doo. Back to this picture again. And again, here you can see, here's this Eustachian tube. Where's my annotate? Oh, I lost my little annotation thing, dagnabbit. Let's just say, you can see my little mouse here. Basically, the Eustachian tube, and it goes all the way to the back of the throat. If you, in fact, maybe I should go back. Let me go back to stop sharing here. When the air pressure is getting higher, you basically are going to have to open up that little tube and make sure that you equalize the pressure from the other side, or else the eardrum will be under pressure. You can feel it yourself. There's this thing called the Valsalva Maneuver. If you like hold your nose and you blow, like don't blow super, super hard, but blow, you'll feel your eardrums pooch out. That's because you're building up the pressure in the back of your nasopharynx, which is going up your Eustachian tube and pooching out your eardrums. And then when you swallow, that tube kind of relaxes, and then you have the pressure equalize again. So I would like everybody, even if I don't see your image, I'm going to trust that you're going to hold your nose and blow, and you're going to feel your eardrums pooch out. That's shoving air up the Eustachian tube into your middle ear, kind of pushing your eardrums out. And then when you swallow, that's going to kind of let that tube relax, open up, and equalize the pressure back. Now this is why if you have a bad cold and there's a lot of inflammation and that Eustachian tube doesn't open, as the pressure gets, either if you're in an airplane, the pressure is getting less and less, and your eardrums might explode. Or if you're diving, if people dive, you probably notice the deeper you go, once you're like 5-6 feet underwater, you have to start hold your nose and blow, increase the pressure inside your middle ear as the water pressure gets stronger. If you can't do that, it gets more and more painful until you actually do burst your eardrums and implode. Like you can't go scuba diving if you got a bad cold, because you don't have the ability to equalize the pressure between your inner ear and outside of your ear. So that is the middle ear. There's more we can talk about it. Clinically, the middle ear is a place that comes up a lot, like in ear infections. It gets filled with either fluids or pus and things like that. So as you continue on in your kind of more clinical studies, you'll spend lots of time. Again, otitis media is like inflammation of the middle ear. Otic meaning the ear media, the middle part of the ear. So I'm sure you will learn much more about the middle ear when you start looking at little kids who have like ear infections. They're pretty common.
So if there's more pressure from the outside of the ear, then you need to open up the Eustachian tube?
In either case, you have to open up the Eustachian tube.
Okay.
If there's less pressure or more pressure, you still need to equalize the pressure. So when you open up that Eustachian tube, your base, in fact, let me go back to my share.
So is it pretty much always open unless you have like a cold or something?
No, it's typically, it typically kind of flops shut. Ooh, I feel frozen. Oh, no, it's back. It typically flops shut, and then when you kind of yawn or swallow, it kind of stretches and equalizes and then kind of flops shut again. So that's kind of why, you know, people chew gum while they're on a plane, or what, if you're driving up to Tahoe. I don't know if I never knew why it was the right thing to do, but I knew as a little kid going up on the vacations at Tahoe, always like, have some gum. So you keep chewing, and every time you're swallowing, it keeps equalizing the pressure. As the air pressure gets less and less as you go to higher and higher altitudes, every time you're swallowing, as you're chewing your gum, that little tube kind of stretches open, equalizes the pressure again, so your eardrum never gets stressed and you're comfortable. So let me, oh, I know what I'm going to do. Yes. Okay. Like I found my drawing thing again. So again, here, Eustachian tube. This is just kind of in the back of the throat, coming up to here. Here's just the outside air coming out to here. So no matter what, whether the outside is getting higher pressure or lower pressure, you just need to make sure you open up this. Basically, you know, the outside air is going through your mouth into your throat, and as long as you open up the Eustachian tube, it's going to equalize, and now you have the same pressure on both sides. But again, the Eustachian tube typically is kind of flopped shut, so it's like kind of yawning or swallowing. It kind of stretches open and makes sure that you have the ability for the air to equalize on either side. All right, so we are going to continue with the ear. So let me go share my screen, annotate. Yeah, I'm going to get better and better at this as we go. So we've been looking at the middle ear here, which rhymes even, and the middle ear is going to be how we're going to transfer the vibration from the sound waves in the air into the fluid world of the inner ear. So the rest of what we're going to talk about now is the inner ear. And again, the inner ear is doing way more than just hearing. The inner ear is also doing your balance, your which way is gravity, which way is your head spinning. So we're going to look at all of that. And let me now look at the picture. No, I just, hold on a sec. I just got an extra, here it is. All right. This picture here is showing the inner ear in more detail. And if you look, there are three main parts, three main areas. We're going to look at each one in turn. This area that looks kind of like these Mickey Mouse ears coming up off the top, those are going to be the semicircular canals. On keyboard. Canals There's 3 of them actually. And they're going to be doing what we call dynamic equilibrium. Sometimes they call it rotational equilibrium. Oh, that should be a different color though. Fuck a duck. Anyway, so semicircular canals are doing this dynamic equilibrium rotational. Which way is your head spinning? Let's make it a different color. Another part is this part right here, which we often call the vestibule. And this is going to contain a saccule and a utricle, which are going to do what's called static equilibrium. Sometimes people also think about them as detecting linear acceleration. Well, that'll all make more sense. We'll look at that in more detail. And then the cochlea, this is the part that's going to be doing hearing, as we normally think about it. So we're going to look at each of these 3 different regions here. And let's start with the semicircular canals. So the semicircular canals, dynamic equilibrium. Basic idea is, in fact, this is where it's going to be useful getting the PowerPoint. I'm going to share the PowerPoint from the textbook here. So the semicircular canals are, let's see, slideshow from current slide. Semicircular canals, there's 3 of them. Each one is kind of like a semicircular canal with this little region here, which is going to be where the hair cells are. So again, we have a semicircular canal, there's 3 of them, and then this little area where the hair cells is called the ampulla. So the basic idea here is there's basically going to be a semicircular canal. And again, there's 3 of them. You have them in kind of the 3 different axes. Again, for like rotating your body, for yaw, or, pitching, rolling back and forth, for pitching back and forth. There's pitch, yaw, and roll. If you were an airplane, those would be your 3 axes. There's the little hair cells in here. Again, hair cells, which are the things that detect bending and turn it into an electrical signal. So off of the bottom of the hair cells is where you're going to just have, the nerve going to send the message that tells the brain what the hair cells are doing. This whole space in here is filled with fluid. This is filled with this fluid called endolymph. Then the last piece we need, there's this little gelatinous membrane right above the hair cells called the cupula. Cupula, which is basically a gelatinous membrane. And the thing that you need to remember is that this is all within the temporal bone. This is your skull. So this is a little cave within your skull. So if you are moving your head, you're rotating. Let's say you're saying no, no, yes, yes, whatever you're doing with your head. If you're rotating your head, this whole structure is rotating. However, the endolymph, the fluid inside here, has inertia, meaning it tends to stay at rest. Newton's law, like motions at rest tend to stay at rest. Normal, if, I'm going to do my best. Normally, if we were in class, I would be able to show you back. I'm going to try to do this. Again, I'm going to get rid of the background so I can kind of try to show this basic idea. Like, here I have a cup. I'm trying to do with, you can actually see there's a black bottom, and this cup has inertia. It's a classic waiter yanking the tablecloth out from under the place setting. If I pull back, watch what I'm doing here. If I pull the paper fast, the cup still stays still. It doesn't move with the paper. That's kind of inertia. So if this paper was like your skull and the cup is like the fluid inside the canal, your skull is moving, but the fluid is not moving. So that's going to be critical here. So I don't know, did people actually see my little picture, or should I share that on a bigger picture?
I saw it.
Okay. So when you are moving your head, the fluid stays still. And if the fluid stays still, but the head is moving, this cupula is like, if let's say your head is rotating this way, the fluid stays still. It's going to be like the fluid is pushing and bending the cupula the other way, in which case the hair bundles are moving. Again, this is actually, it's also, again, it's worth kind of getting, I'm going to stop sharing for a moment. Like if I have, imagine water instead of the endolymph, air. Air is a fluid. Let's say this is the cupula. If my whole skull is moving one direction, then the cupula gets bent the other direction. Like I just made an arrow so you could see, like if I'm going, my body is going this way, but the paper bent the other way because it hit the fluid, the air, which pushed it the other way. If my body is going to go this way, boom, then you just saw the membrane actually bended the opposite direction. So basically, whenever you're spinning your head around, you are going to be whenever you spin your head around, you're going to bend the little hair cells that are there at the base of the cupula. Again, if you spin your head that way, the cupula gets bent that way. That's going to send a message to your brain that your head is spinning. And again, because you have 3 of these in the 3 different, like if you, again, I can show one more thing. Like I'm going to stop sharing this again. Right. You have, they're not exactly, but ones that can detect if your head is rolling. One can detect if your head is pitching. One can detect if your head is yawing. You have 3 semicircular canals, so you can detect your head rotating in the 3 axes.
So the fluid pushes against the cupula, which then presses against the hair cells?
Exactly. The other thing we can add in here is right? I kind of showed if I kind of pull this paper quick, the glass doesn't move. But it is possible if I kind of just keep things moving, the glass and the paper are moving together. So like if, like what's like a class, this is harder to do when, again, when I'm not in the room, but I could start, you could probably maybe, can you hear me when I'm sitting back like this?
Yeah.
Yes? So if I start spinning right now, at some point, my skull and those fluids in my inner ear, the endolymph, they're both moving together. So right now, I'm actually spinning. Can you all, let me, whoa, can you all see that I'm spinning?
Yes.
And I'm not feeling dizzy. Why do I not feel dizzy?
Because the endolymph is rotating at the same speed as your skull.
Exactly. So the endolymph and the skull are moving together, which means there's no bending on the cupula. So I don't get any sense of being dizzy. I mean, I see the world spinning weird, but I don't feel dizzy. What happens if I stop spinning all of a sudden? Think about inertia. If the fluid is spinning with around and around and I stop spinning really quick, what's going to happen?
Gonna bend everything quickly.
Yeah, the fluid will keep moving, but the skull stops.
Just fantastic.
What's
2
that?
more hours of this.
All right, so I want you all to look at my eyes. Can you see my eyes?
Exactly.
Did anybody see my eyes?
So weird.
Megan, we can hear you.
Yeah, Megan, you're kind of distracting.
Yeah.
She's still talking.
Thank you. There's actually probably more than 2 more hours of this, 2 hours and 15 minutes. But so did anybody notice my eyes? I don't know if you could see.
They were twitching back and forth.
They were like twitching back and forth. That's actually something that is used clinically. It's called nystagmus. If your inner ear is sending messages to your brain that you are moving, right? Normally there's this reflex. So I can like get back to normal here. La la la. Let's go back into just the aurora. If my brain is, or if my head is moving, let's say I want to keep looking straight ahead. If I turn my head one direction, there's a reflex that has to move my eyes to this side. So I keep looking. If I want to keep looking straight ahead and I move my head this way, there's a reflex that's pulling my eyeball the other way inside my skull. So if I'm moving my head, I gotta be moving my eyes back and forth if I want to keep looking straight ahead. If my brain thinks I'm spinning, even though I'm not, because that fluid is still got motion from my current, like spinning around, my brain is thinking, oh, he's spinning. I better move his eyeballs to make sure he still stays looking ahead. But in fact, what you see is just the eyes starting to flick around as the person tries to look straight ahead. So nystagmus, let me write that up on the board here. That is that condition when your eyes are kind of flicking because you are trying to look straight ahead, but your brain thinks your head is actually turning. You can get it if you have different different ear conditions. people who have like labyrinthitis and stuff, things where their brain is constantly getting messages that they're turning, even though they're not, their eyes are going to flick. If you want to make it happen in a clinical setting, you can actually just pour warm water in somebody's ear. They call it caloric nystagmus, because when you put the warm water in there, the water, that warming things up creates these convection currents. The fluid starts flowing a bit, pushes on the cupula, pushes on the hair cells, and your brain thinks you're turning, and then your eyes start moving. So nystagmus is something you should do it at home. You can actually, it's fun. Have somebody spin around and around and around and around, and then have them stop and look straight at you and watch their eyes. And it's really kind of fun and hilarious to watch their eyes spinning back and forth. So
you were saying that the fluid has inertia, but I guess when you're spinning, the fluid moves. It's just when you tilt your head, it doesn't move.
So the fluid typically has inertia, but when you just keep going and going, then eventually the fluid starts moving along with your skull.
Okay.
So, like for instance, if I'm going to look really ridiculous doing this, but I can show you the concept, if not quite the practice. Like if I was in a ballet and I wanted to, I got to turn off this sharing thing in the screen. If you're in a ballet, you do these crazy spins around and around. You don't want to stop and then go, whoa, and like fall on your face when you're supposed to do your next little deal. So how can you, if you're a dancer, spin around and look super cool and then stop spinning and then go do whatever you're going to do without falling on your face?
Your eyes are focused at one point.
Yeah, they do what's called spotting. Kind of the same thing where if I had my cup and as long as I pulled this paper quick, the cup didn't move. But when I kind of kept pulling it more slowly, the cup started moving along with it. If you're spotting while you're doing like a ballet dance, you're going to turn your body, but keep your head looking straight ahead. And in one really fast snap, bring your head back around. So your body is kind of spinning, but you try as much as possible to not have your head spin and have your head only move in this really fast snap, which makes sure that the fluid never gets entrained and starts rotating with your body. So then when I'm done with my spinning, I come out of it, I'm like not dizzy at all. I can go and do my next fancy part of my dance. So the fluid typically does have inertia for normal things, but if you keep moving around and around and around, eventually it does get caught up with the motion, and then that's why you do get dizzy. So let us continue with our discussions here. All right. So, any questions about semicircular canals and this idea of, we call dynamic equilibrium because it's kind of what's your body doing as it's moving, or your head, I should say, or it's also called rotational equilibrium or rotational acceleration because it's telling you what's the rotational acceleration of your skull.
Okay. But then when you just tilt your head like sideways, the fluid does not move, right? But you still get that signal.
Yeah, the fluid does not move, but my skull moves, and that's what's making the cupula bend, and that's what's telling my brain that my head is rotating.
Okay.
Yeah, exactly. All right, so question?
What happens with vertigo then? If someone always has vertigo, is the fluid just still moving?
Yeah, or there's some kind of inflammation that's putting pressure and keeps the hair cells bent.
Okay, got it. So
Yeah.
like, what could cause that? Could that be like a number of things,
Oh,
dysfunction?
so like ear infections, for instance, can do that. When you get inflammation, I can say I had an ear infection once where I just felt dizzy for like days. I kind of felt like car sickness is often when you have a disconnect between what you see and what your equilibrium senses are telling you. So I would like walk around the classroom and have to like try hard not to just barf on people because I just was always feeling dizzy and nauseous and stuff like that. So ear infections can do that to you. There's a disease that I don't know if they know what causes it, but it can be really debilitating called Meniere's disease. And those people, there's something goes wrong in the kind of operation of this, and people just feel kind of off balance and kind of car sick all the time. So different things. There's labyrinthitis. There's some conditions that are more chronic and that people just have to try to learn to live with and medicate. Other ones that are more just acute because they've got like an ear infection or something. So, but yeah, it can be incredibly debilitating. And these reflexes tend to get slower and not work as well as you get older. So older people have worse balance, more likely to fall over. A lot of times, people who are older, they do a lot more exercises trying to maintain their balance because you can't just assume it's all working well as you get older. I should stop saying because now everything's getting recorded. Damn. I gotta learn how to be a like YouTube star. There, I just said again. The next part that we're going to look at is the part that lets you know which way is gravity pulling. So if we go back, share screen. What did I just do here? I'm going to share screen and go back into here. So everything we've been talking about right now has been in the semicircular canals, which I kind of drew up in red there. Now I'm going to do some things. I'll do them in fuchsia. So this part right here that I'm kind of drawing in fuchsia, which we call the vestibule, is the utricle and the saccule. So the utricle and the saccule, which are in this area we call the vestibule of the inner ear. The basic idea here is still you're my good friend, the hair cells. They're going to still be the major players, but now we're going to connect them in a really different way. So what I'm going to describe right now is the utricle and the saccule. The only difference between them is they are set at perpendicular orientations, so they can detect the pull of gravity from different orientations. So these basically tell you which way gravity is pulling. Sometimes we call this static equilibrium. And again, you still have, this is the skull. Here's the hair cells. The hair cells are kind of in a little patch they call the macula. macula just means spot. So it's like a little lawn of hair cells. And then sitting on top of the hair cells is this big gelatinous thing filled with calcium carbonate crystals called otoliths. Oto means ear, liths mean rock. It means basically ear rocks. So these are little calcium carbonate crystals. I'm going to call them like rocks. So you basically have hair cells with a bag of rocks on top. And again, it's anchored to your skull. So as you move your skull with respect to gravity, these rocks are going to get pulled by gravity. So if, let's say this is your skull, here's my little hair cells, here are the rocks. They're going to get pulled due to gravity. This is down, this is up. The hair cells are going to get bent this way. If you tilt your head the other way, the hair cells are going to get bent the other way. So whichever way you tilt your head, it's going to change the direction of bending on the hair cells because they're getting bent by the weight of this otolith, these little rocks, which are getting pulled by gravity. And so this is how your brain is getting information about which direction is gravity pulling. Go back to the PowerPoint thing. So if we, so for the semicircular canals, it was about the spinning of your head. But now when we go to the little otoliths in the saccular macula and the utricular macula, as you tilt your head with respect to gravity, the little stones are going to get pulled. Again, as he's tilting his head forward, these little stones get pulled by the gravity, bend the little hair bundles, which creates an electrical signal and tells your brain, oh, he's tilting his head down compared with respect to gravity. Whereas when his head was with this angle with respect to gravity, it didn't bend them. So then the brain knows that it's a different situation. So the sacculus and the utriculus, or the saccule and the utricle, they're synonyms, are basically ways of telling which way is gravity pulling. We also sometimes consider this linear acceleration. Because basically what you're detecting gravity is just acceleration 9.8 meters per second down.
Absolutely.
But linear acceleration, if he's speeding up here, not if he's at a constant velocity, but if he's actually accelerating forward, that's also going to cause this bending of the hair cells. So if you're in your sports car and your Tesla, if you've ever been in a Tesla, it's freaking crazy. You like hit the, I have a friend did this, you like hit the gas, not even gas, you hit the gas pedal and it just starts accelerating faster and faster and faster and faster and you're plastered behind the seat. That feeling of accelerating faster and faster and faster is going to cause this same thing. So you can think of the saccule and the utricle also measuring linear acceleration. Or you can also think about them just telling you which way is gravity pulling. So somebody just had a question? Any questions?
Dr. Eggert, can these little gelatinous things rupture?
I think what happens is sometimes they get stuck. Like for some people, they like stop moving, and then there's ways to try to loosen it up so then the system starts moving again. But it can get stuck, so you keep getting a signal that gravity is going the wrong direction. So that is something that can happen, I've heard of. I don't know a lot about the more clinical sides of this kind of stuff, though. You probably want to talk to like an ENT about that kind of stuff.
Cool, thanks.
So there you've got the equilibrium senses. And let me go back to, I'm in the right spot. So if I go both, even though officially, let me kind of clear this off a little bit here. Annotate and clear. Oh, I'm in the wrong thing. Officially, vestibular is utricle and saccule, but in reality, when people talk about the vestibular senses, they usually actually mean both the semicircular canals and the utricle and the saccule. Those are your senses of equilibrium. And sometimes people use vestibular senses and equilibrium senses as kind of synonymous. Again, even though officially semicircular canals come off of the vestibule, don't get too pedantic about it. Usually people just talk about collectively the vestibular senses, utricle, saccule, semicircular canals. So now if we can move on to the final part of the ear, and actually our final part of the nervous system, which is hearing, which is happening in the cochlea. So let's do this. Oh, I see Allison is here now. Allison Orozco got here. All right. All right, so the cochlea, again, looks kind of like a snail. I haven't been talking so much about this idea of the bony labyrinth and the let me go back here. In general, you have what's called the bony labyrinth, which is this cave that's in your ear that's made by the bone. And then there's what we call the membranous labyrinth, which is this inner part, which is inside kind of a membrane. And there are these two kinds of fluids that are going to be important. So far, we've just been talking about the idea of endolymph, which is in the membranous labyrinth. But then there's also a different fluid that's in between the membranous and bony labyrinth called the perilymph. Again, don't get too concerned about the differences between their compositions and how that. Mainly what you want to be aware of here is when you have sound waves coming in, gosh darn it, sound waves coming in, vibrating the eardrum. Eardrum pushes and pulls on the ossicles, these little bones, which vibrates the oval window. We talked about the oval window being the entrance into the inner ear. That's going to set up these pressure waves in the perilymph that are going to travel up one side of this tunnel made between the bony and membranous labyrinth here. This is actually going to be called the tympanic duct. In this picture here, this is supposed to be the cochlea, but it's been a little unwound. Normally the cochlea has 2.5 turns, but this is pretend you were unwinding it to make it easier to draw. So the vibration that starts as an air vibration becomes a fluid vibration that goes up one side, goes around the tip of the snail, and then comes back the other side. And then the round window was that place I said where the pressure release can happen. So things to think about in here. So again, the vibration starts by going up one side of the cochlea, this tunnel here, which is called the tympanic duct. Can I add text? Tympanic duct. This helicotrema, that's like the very tip of the snail. That's where the signal kind of comes back down the other, which is going to be called, they're saying scala tympani. You can also call it tympanic duct.
I'm a little bit confused. So the pressure after going through the oval window, it becomes a fluid vibration, then it just travels through the perilymph and the tympanic duct.
Yes. And again, if we were in a real board and I could, this is actually, this is a really awkward way for me to try to explain this. But basically the vibration that's an air vibration becomes a fluid vibration, which, let me kind of go back here. Back here even, this is air vibration transformed through the ossicles into a fluid vibration, and then it's going to go up one side of the snail, the vestibular duct, and then go at the helicotrema, make a U-turn and come back the other side, which is the tympanic duct, and then pressure release at the round window. If that makes it make any more sense there.
So do we need to know the process? So after it goes to the oval window?
Yeah, you should know that once it goes to the oval window, then a fluid wave is going to go up the vestibular duct, loop around at the helicotrema, and then come back down the tympanic duct, and then release at the round window. And what's going to happen that's going to be interesting is in the middle, which is this cochlear duct in the middle here, which is where we have the hair cells with the, that are going to actually detect that movement, that vibration, and actually convert it into an electrical signal, which the brain is going to be able to get. So far, all we've talked about is this fluid wave going up one side and back the other, but this duct that's in the middle, this cochlear duct, this is where the action happens. This is where you actually get the conversion of vibration into an electrical signal. So let me pick a good color. Maybe I'll pick violet. Here, right along this division here that I'm just drawing in this kind of lilac between this tympanic duct and this little blue cochlear duct. This is this membrane called the basilar membrane. Its name is right here, the basilar membrane. The basilar membrane is going to be super important for two reasons. One reason is, so here's the basilar membrane again, if we did a cross section through here. So we've got the tympanic duct here. So the sound's coming in here, kind of coming back this way. Sitting on top of this basilar membrane is what we call the organ of Corti. Organ of Corti is basically hair cells with this little membrane over the top of it. So as the basilar membrane vibrates, as it vibrates up and down, as this pressure wave is moving through, basilar membrane moves up and down. Hair cells with this tectorial membrane, they're going to wiggle, and that's going to create electrical signal. In fact, let me go back to my little annotating thing. I think I can make a stamp. I'm going to make little red stars. It's like bing, bing, bing. Or can I make them red? Maybe not. Oh, they're just so. Basically, as the basilar membrane moves up and down, then this organ of Corti is going to wiggle, and the organ of Corti has the hair cells, which means we're converting electrical signal, motion into an electrical signal. Again, if you're thinking about sensory transduction of hearing, if you're thinking about stimulus going to an electrical signal, stimulus starts out as just sound in the air, which then vibrates the eardrum, which then is transferred through these little ossicles into a vibration of the oval window, which creates a fluid vibration in the vestibular duct and back down the tympanic duct, which then causes bending of the basilar membrane. The basilar membrane bends. That means the organ of Corti with its hair cells is moving. As those hair cells are bending, it creates an electrical signal that goes down your auditory nerve to your brain. So you should, each of those things I just said should make sense. Maybe I'll type it out in just a few moments. The other part of this that we need to add in fact, let me do it just on a whiteboard because I think it's going to be less awkward. So here we had the oval window, where basically the vibration is entering, coming into our tympanic duct here. The fluid looping around at the helicotrema.
Wait, does it go to the vestibular duct first or the tympanic duct?
Like I said, it goes into the vestibular duct. Maybe I'll have to go into my Premiere and we'll never know. I'll just rewrite history. I just wrote vestibular duct to start with. Comes back along the tympanic duct. And in the middle here is the cochlear duct. And this is where you have the hair cells and the organ of Corti. This is where you actually have the stuff that can convert vibration into electrical signal. And the membrane that the organ of Corti is sitting on top of here, this is called the basilar membrane. And the basilar membrane has very different qualities. In fact, hold on one sec. All right, so like here, this is a guitar. If I go like to the low E string, it's very kind of big, and it's kind of wound with extra wires around, so it's kind of big and heavy, and its natural frequency that it vibrates at is kind of lower. On the other side of their, God, I wish, there you can see. This is a much smaller, thinner, more tightly wound, tight string. Its natural vibration is much higher. This kind of thick string has a natural vibration that's much lower. So just the mechanical qualities of something can determine its resonance frequency. At what frequency does it want to kind of vibrate at? In the basilar membrane, the part that's more near the oval window and the round window is here as well. The part of the basilar membrane that's down here responds to much higher frequencies. The part of the basilar membrane that's up near the helicotrema responds to low frequencies. And the mechanical resonance of a structure, it's not just like if you pluck it where it vibrates, it's also where it absorbs energy. If you remember, there were those commercials like, is it live or is it Memorex? There'd be like some opera singer and she'd go, and then all of a sudden the glass would explode. You've probably seen that kind of thing before. But basically, if you have a really, in fact, again, here is a glass. If I flick it, it has its resonant frequency. The frequency it likes to vibrate at if you thump it. It's also where it will absorb energy. Like, if I could make a sound really loud at that same frequency, it would be absorbing that energy and taking it in. That's how those opera singers explode the fine crystal glasses. It's the same kind of thing in your inner ear here, though. When a sound is coming in at a certain frequency, it's going to be absorbed and shake this basilar membrane at a different part depending on what the frequency is. If it's like low frequency, it's going to be vibrating down here, and means the hair cells in the organ of Corti over on that part of the cochlea are going to get shaken and send a message to the brain saying, oh, it's a lower frequency. If it's a high frequency, then you're going to have the basilar membrane absorbing it and shaking closer to the oval window part here, and it's going to then shake up the organ of Corti and the hair cells that are over on this part of the cochlea, and those hair cells will depolarize and send a message to your brain, so your brain knows that it's a higher pitch sound. So that is kind of a first approximation of how your brain can get information about the frequency of the sound, depending on if it's a lower frequency vibrating the organ of Corti down more to the helicotrema, high frequencies vibrating the organ of Corti up near the other side. So your brain gets information about the frequency. Does that make sense?
Once the fluid vibration goes to the tympanic duct, it just exits out the round window?
Yes.
And then what? And it's just like gone?
Yeah, it just dissipates.
Okay.
In reality, if you wanted to get into the more nuanced physics of it, you actually kind of have the energy kind of short circuit across Doesn't necessarily go all the way up to the helicotrema, but it doesn't matter so much. This is like if you have a cochlear implant. A cochlear implant is for somebody who has sensory neural deafness, right? The system isn't working. You can actually take an electrode array. Let me do this. I will. And you basically thread it up the cochlear duct. And then you have electrodes that you can turn on at different regions along the way. So this is what I'm drawing now is like, this would be what a cochlear implant is. So this electrode array right here that I've just put in. And basically you have like a little microphone that's just listening to the sound out in the world. And then there's some little computer that analyzes the frequency content of the sound and then activates this electrode array appropriately. So whatever the frequencies, so this is kind of frequency analyzing. Analyzes the frequency of the sound in the room and will turn on these electrodes appropriately to stimulate the cochlea. So it should, like it would be if the system had been working properly. You know, obviously, this is not going to bring back normal hearing. Somebody who gets this as an adult, it will give them more information than they had, but it's still not going to just sound like your hearing's back. For small kids, where there's still a lot of neuroplasticity, there is evidence, like if people get these cochlear implants when they're still very young, they can actually have pretty usable hearing just using this system. If you get it after your auditory system is kind of more developed, it's going to give you information that's useful, but it's not going to like restore your hearing like it used to be or anything. But that's all a cochlear implant is that. The reality is there's a little more going on in terms of the way the ear gets sound or analyzes sound. Let me look at the time. I'm just going to spend a couple of moments talking about this. So some closing thoughts on hearing. Partly because this is what I did all my work. So there are different ways. How do you tell where? So signal from the noise. Let's talk about signal versus noise. You know, in the world, there is all sorts of noise, auditory noise. There's whoosh, there's wind going and crackling branches and who knows what. And then there's also the noise just of your blood rushing through your head and the vibration of your muscles. And so there's all sorts of noise. This is inside and outside. Inside your body, all of just like, again, the rushing of the blood. Outside, the wind and the whatever. Signal is there's a tiger trying to sneak up on you and you better notice that, you know, maybe it just stepped on a branch and the branch went crack. And it's like, oh my God, I gotta look because there's some danger here and I don't want to be a tiger's like midday snack. So there's a couple of ways that you can try to figure out signal from noise. One of them we have actually kind of looked at a bit in our lab, I'm going to say, so both of these are going to depend on both frequency information and timing information. Tympani. So back in this thing I was just talking about over here, this is about how you can detect frequency information of the sound coming in. I'm not going to get into the detail of it now, but the ear is actually way more complicated than just this, because it's also good at really preserving the timing information. When does the sound start? When does it stop? If you have a system that is just working on these physical resonances like I was talking about, it's not very good at preserving timing information. There's actually parts of the organ of Corti that are efferent. There's some of those hair cells, what we call the outer hair cells, actually do the opposite of what we've been talking about. We've all been saying hair cells, oh, you bend the hair cells and then it sends an electrical signal to the brain. But a bunch of the hair cells in the organ of Corti do the opposite. The brain sends an electrical signal to them, and they actually physically move and change the tuning properties of the ear. Again, I'm not going to get into the details of this because we don't have time. I will say one of the weird things, because those some of the hair cells, those outer hair cells, are actually physically moving, they're actually setting up their own vibrations due to their motion, which then actually vibrates the round window, which ultimately through the ossicles vibrates your eardrum. So you have what are called otoacoustic emissions. You can actually put a very sensitive microphone by your eardrum, and you can actually hear weird little sound coming out of your own ear, because those little hair cells that are actually actively moving to do this more fancy timing information stuff are actually causing their own little signal that you can hear coming out of the eardrum, which is kind of fascinating. But back to our separating signal from noise, frequency information, and timing information. Let's just end on this. I'm going to talk about how can you use frequency information to find a signal amidst the noise. So using frequency info. So if you think about the frequency spectrum of the sound, from kind of low frequencies to high frequencies. So there's, let's just call these bins. Let's look at the sound energy that's at the really low. Sound energy that's really high. In general, a lot of the noise that we don't care about, like wind and stuff, what we call like white noise, it's kind of equally spread out. Its energy is a lot. It might be a lot of it, but there's a lot of it in the low and the mids and the highs. There's high and low. So you don't see it really focused in any particular frequency band. So a lot of noises tend to be more kind of evenly distributed. Whereas something like a snapping branch, when a tiger is trying to sneak up on you, it is often going to be a lot of energy in one very narrow frequency band. So like that snapping stick, high frequency, so maybe I'm going to do, so here's like, stick snap. it might not be all that much energy in the big picture. It might just be a little bit compared to the overall energy of all of the white noise from the wind. But because the wind is spread out in all these different bins, you can actually notice, like, okay, this wasn't a whole lot of energy, extra noise, sound energy compared to all the other. But if I have the ability to look just at the noise, the sound energy in this one little frequency band, I can say, wow, a whole lot of energy just showed up in this little narrow thing. I want to pay attention to it. Something happened. So this is one way you can separate a signal out of the noise. A lot of the noise tends to be spread out. Even if it's a lot of energy overall, it's spread out across the different frequencies. Whereas a lot of times the particular signal that's interesting that you want to pay attention to, like that tiger walking and snapping the stick, maybe it's not as much energy overall, but it's really concentrated in a narrow frequency band. And if you have the ability to do this fine frequency discrimination, you notice it. And obviously that's a good thing. Does that make sense? You can use, by having this ability to have this fine frequency discrimination, you can notice something happening in a very narrow band of frequency, even if the overall amount of sound energy is really low compared to overall, but you can still notice this interesting thing. That makes sense? Yeah? No? How about do the little thumbs up if you get it. Click on the little like thumb. Okay, cool. Woo! All right, and the last part here, using timing information to detect signal from noise. All right, so this you have actually seen in lab. So like here's you with your ears. And here's just kind of space. And again, maybe this tiger is walking on the stick here. Maybe it's just one little bit of noise amidst all the noise that's happening. But because you have timing information, we saw that using the timing difference between the two ears, you could actually use that for sound localization. So you can actually just think about what is the sound that's happening from this area of space, because there's a certain timing difference between the ears. Or you can look at just the sound appearing from this part of space, this little sector, because that's going to have a different timing difference between the two ears. Or let's look at this part of space. Or let's look at, this part of space. So even though this tiger here is making a noise that's not that loud compared to all the other noise, in this little slice that you're looking at, you can see, oh, something appeared here that wasn't wasn't over here. So timing information lets you also find a signal out of the noise, but rather than by looking for sound energy in a particular frequency band, you're looking for sound energy in a particular kind of spatial area that you can discriminate, because the timing difference lets you kind of detect things spatially.
So are you saying that if you're like facing the tiger, it'll take less time to like hear the noise than if you were facing away from it?
It's not going to take you less time, right? Sound is basically getting to your ear at the same speed.
So what do you mean by timing difference?
Oh, the timing difference, if you remember, how can you tell? Remember when if we just had the person here and we just rang the bell. I don't know if you remember, we rang the little bell. How could you tell if they rang the bell on the right side versus the left side if your eyes were closed? Do you remember? We did this in lab. I'm pretty sure didn't we do this part in lab? Anybody remember this?
I don't think we did that.
Oh, maybe. Oh, then okay. This is something maybe I'll set up for you to do with your at home with your, or you could do it with yourself if you're sheltering in place by yourself. No, you can't do it. Shit, I'll figure this out. Basically, this idea of interoral timing difference. So sound is actually pretty slow. If you've ever seen, lightning flashes and like 5 seconds later, you can hear the thunder. So you have very low speed of, compared, normally your nervous system is super pokey, but in the terms of sound waves, sound is slow enough that if the bell is over here, this ear will actually hear it before this ear hears it. So your brain can use that difference to know, oh, it must be on this side of my body because this ear heard it first. If I had a bell ringing on this side, it would get to this ear before this ear. So then my brain has, using this timing difference between the two ears, it would know, oh, this sound is coming from that side instead of from that side. So this is one of the ways your body knows, like, where in space is the sound coming from. So that is what I mean by this. By looking at the timing difference between the two ears, you can tell what part of space the sound is coming from. Does that make more sense now?
Mm-hmm.
Okay.